All right, folks, I just heard my voice say that I will talk to you in about 45 seconds here, which means we are live all over the globe right now, and there's no better way to be spending a Tuesday morning or a Tuesday afternoon, depending on where you are located. And this is a live session, so where you are, we would love to hear if you can drop in the comments where you are watching from. I know the team would be really excited. And the good news is that you are in the right place. I see that we've got already 15, 16, 17 individuals joining with us. You are in the right spot. This is a LinkedIn Live hosted by Upperton Pharma Solutions, the use of spray drying for oral and nasal drug delivery. We're going to talk about approaches and challenges and have a really fantastic group of panelists here of experts we're going to just jump in. We're going to answer your questions. We've got an outline that we're going to work through that's going to address some of the critical elements of the conversation, but we also want to hear from you. So if you have any specific questions for any of the panelists, you are absolutely welcome to fire that into the comments, and I will ask that of the team. And I know we are just a couple of minutes into this, and we're at 20 people here and growing, and so I do want to go ahead and get started. I'll do another little overview after introductions, but the less that I talk, the better, which means we're going to have our group of experts introduce themselves. And up first on my screen, Richard Johnson, I'm going to pass it to you for an introduction.
Hello there. Thank you very much. Hello, everybody. My name is Richard Johnson, and I'm the founder and chief scientific officer at Upperton Pharma Solutions. So by definition, I've been at Upperton since I formed the business, which is now 26 years ago. My primary interest is in spray drying and pharmaceutical development. I've been involved in the spray drying of a wide range of molecules, from small molecules to much larger biologics, to make a really wide range of dosage forms, from tablets and capsules through to pulmonary and nasal products, and now more recently sterile injectable products. So I have a long history and a broad background in the use of spray drying to make pharmaceutical products.
I love it. I love it. Richard, we're really thankful to have you on the call today, and I'm looking forward to hearing from your expertise as well. Jack, I'll toss it to you.
Yep, hi there. I'm Jack Sorrell. I'm a development manager in the R&D department at Upperton. I've been at Upperton for around 10 years now, and like Richard, have extensive spray drying experience in both bioavailability enhancement and also particle engineering for both inhaled and nasal applications too. So yeah, I'm looking forward to discussing all things spray drying.
Awesome. I love it. Laura, that means you're up next.
Yeah, hi there. I'm Laura Mason. I'm Director of Pharmaceutical Sciences here at Upperton. I've been working in the pharmaceutical industry for the last 15 years. I'm a pharmacist by background and a PhD in solid oral dosage form development. So I guess my passion is in actual getting those drug products through to clinic. I've been at Upperton now 8.5 years, heading up the R&D team. So seeing the hundreds of different projects we've had coming through. So looking forward to chatting to everyone today.
Wonderful. Thank you so much for spending an hour with us. And that means Richard Weaver, you're up next, saving the best for last. Why don't you give us an introduction here?
Yeah, thanks a lot, Alex. Yeah, I'm not sure about that, by the way, but yeah, thanks. Yeah, so I'm Richard Weaver. I'm the managing director and founder of a company called Drug Discovery Solutions. We offer consultancy in the DMPK and ADME space. Rewinding a little bit and my interactions with Upperton. So I'm actually a chemist by training, a degree and PhD in synthesis and postdoc. I then joined Astra, again showing my age, in 1997, which became AstraZeneca, and I was a DMPK scientist, project scientist there for many years. The site closed and I saw a gap in the market, and I launched my own CRO called Xenergesis, and that was at Nottingham in BioCity, and that's actually where I met Richard for the first time. So I've known Richard and team for a good 10 plus years. That was a wet-based laboratory in the DMPK area, so all things DMPK, and obviously there's a big strong link between DMPK, formulation, bioavailability, predicting with confidence into human. And I guess that's where I got really interested, because at the time, back in the pharma days, there were a lot of kind of you do your bit in DMPK and then throw it over the wall and let the formulation guys get on with it, which is completely wrong. It should be a very joined up process. So this is my passion, to be honest, and why I'm delighted to be invited by the team. We've worked closely with Upperton, quite a few years ago, done some nice projects together, and yeah, delighted to be invited here today. Thank you.
I love it. I said best for last. You doubted it for a split second, but now I'm fired up, Rich. I think that is an unbelievable way to set the tone. We're 5 minutes into this LinkedIn Live session. I just wanted to reiterate that this is a live conversation. And so if you have questions of the experts on the panel, you can fire them into the comments. I will ask those questions here. And if you're watching this 2, 3, 4 days from now or a couple of weeks from now, but you still have questions for any of the panelists, you can still put those comments into the comment section, and the Upperton team will make sure that we are able to answer them. And again, this is the use of spray drying for oral and nasal drug delivery, where we're going to talk about approaches and challenges. And Richard Johnson, I kind of just want to rip the Band-Aid off and get this conversation started. And so I'm going to ask you, if you can, to just share a high-level overview to spray drying and its value for oral and nasal drug development. I think starting there and then being able to filter through this conversation is a good place to begin.
Yeah, perfect. Yeah, happy to do that. Well, basically, spray drying, as the name it sort of tells you, really, it's all about taking a solution and spraying it or atomizing it into a heated chamber to make dry powders. So it's in many ways, it's a relatively simple drying process. You spray liquids into a heated chamber. Those little droplets of liquid dry, and whatever is in solution then becomes a dry powder. So a very simple process, actually a process that's been with us for well over 150 years, used primarily in the food industry. So as a way of producing large amounts of dried products, so for example, milk and eggs and things like this, things with short shelf lives. So it's a very rapid, very scalable drying process. But there's some unique features about that process that allow us to be able to manipulate spray drying and use it in the pharmaceutical industry. And I think there are a number of key features of spray drying that we can utilize. The first being whatever is in solution, your active drug and the excipients that you put in solution, and that can be an aqueous water-based solution or a solvent-based solution. When you mix those together in solution and spray and dry them, those molecules are in an intimate molecular mix. So it's not like a blend, it's a molecular mix of an active drug with excipients. That's one really important feature. The second important feature about spray drying, or one of the key features, is that ability to make particles, or first of all, droplets of different sizes, and then subsequently particles of different sizes. So we call that particle engineering. And that's really all about making particles of different sizes and being able to then direct where those particles can go. So we can change the spray drying conditions to make big particles or small particles. So coming back to the original question, how is spray drying used in the pharmaceutical industry? Well, for nasal products and also pulmonary products for delivery into the lung, it's all about using spray drying to make particles containing your drug and those excipients of different sizes. That's exactly what you need in this case for nasal delivery, to target into the nasal cavity. So I'll talk a bit more about that later. The second important feature is that ability to make a molecular mix of your drug and an excipient. And that can really change the physical properties of the drug in question. And this can now be used primarily in oral drug delivery, where we're looking to change the physical and chemical or the dissolution properties of that drug by spray drying it to make a different type of particle, a different type of drug mixture that has different physical properties. And I believe Jack will probably talk a bit more about that next.
Yeah. Well, I think, Jack, just to transition right into you, if you want to jump in and follow that, I think it will set up for the rest of the conversation. Absolutely.
Yeah, sure. So I guess to frame it, why is bioavailability enhancement so important? It's generally because so many drugs in the development pipeline now are considered poorly soluble. So some estimates have up to 80% of new development APIs as either BCS class 2, so poorly soluble but good permeability, or BCS class 4, poor solubility and poor permeability. And I guess that's important because if you can't dissolve your API in situ in the body, then it will have poor absorption and ultimately poor efficacy. So spray drying generally improves bioavailability. And one of the main ways we can do this is through the production of amorphous solid dispersions. So these are generally comprised of spray-dried mixes of the previously poorly soluble API alongside polymer excipients such as HPMCAS or PVP or Udrajet. And what happens in these formulations is that the API is trapped within the polymer matrix in an amorphous state, which exists at a higher energy state than the previously sort of more thermodynamically stable crystalline API. And this higher energy state allows the API to dissolve easier in solution at higher levels. So this is known as the spring effect. But secondary to that, the polymer, once the API is dissolved in solution, will then stabilize the API and prevent it from essentially precipitating from its supersaturated state. So this is known as the parachute effect. In terms of the formulations themselves, we typically target between 10 to 50% API loading within these polymer matrices. That loading level is generally a balance between optimization of the API within the polymer to reduce dosage requirements, while still maintaining a ratio at which the polymer can also stabilize the API in solution. So it's rarely a trade-off. As part of testing and sort of development, we would generally look to spray dry the API with a range of polymers, including some of the ones we mentioned earlier. And generally, we would, once spray dry, we would look to characterize those formulations in a variety of ways to determine lead formulations.
Yeah. Well, I'll go ahead, Richard.
Yeah, I was just going to say it's clearly a bit of a balance or a trade-off, as Jack was saying, between wanting as much drug in that spray-dried dispersion as you possibly can from a process point of view, from an efficiency point of view, but still maintaining that amorphous property of the spray-dried powder. So if you have too much drug, it might crystallize out. But also when it goes back into solution, you don't want too much drug present, otherwise it could again start precipitating out if there isn't enough polymer there to then stabilize it. So a bit of a trade-off, as Jack was saying, between getting the best properties of the drug polymer mix, but not putting too much drug in there that you then lose what you're looking for, which is improved solubility and ultimately bioavailability.
Yeah. Well, and I know we're going to talk a little bit about testing and evaluating, but I did get a question already in here, Jack, that I think you are probably best to answer it just based off of that initial setup. And the question here is, can you share some use cases where spray dried dispersions have been especially effective in enhancing poorly soluble molecules?
Yeah, there's actually a really good sort of case that we're working with in the R&D department right now, actually. So
Great.
The API itself is very poorly soluble. So it has less than 1 microgram per mil solubility in, say, a phosphate buffer. We have recently spray dried that API at 10% loading with HPMCAS, and the solubility is now increased to approximately 0.5 megamil in the same solution. So we've had around 1700-fold increase in solubility, in that case specifically. And I guess the next challenge is that formulation as well is now how do we optimize the drug loading within that formulation? Really, as we've already mentioned, to help reduce dosage burden going forward.
Yeah, maybe I can just chip in there as well. That's something that we did. There's quite a few years back now, but when we were at Xenergesis, and we did this with Richard and the team. And again, we can talk about this because it was presented at a conference. A compound called raloxifene, so that has a very low human bioavailability of around 2%, and a lot of that is to do with the poor solubility. We did a spray dried formulation that we tested in vivo preclinically. And we proved that it had, I think it was a 7 or 8-fold increase in AUC, Cmax, and bioavailability. So there was a real example that was essentially recapitulating something from the literature, but real in vivo evidence of the spray drying technique being very successful indeed.
we're not even 15 minutes in, and we're pairing that scientific expertise with some real world experience. So I absolutely love that, Jack. Thank you for sharing that, and Richard for jumping in. I do want to kind of segue here, and Laura, I think I'm going to toss this over to you as we start to talk about the process for testing and evaluating the spray dried prototypes. I know that you and Richard Weaver over here can talk a little bit about the PK studies, but just want to start at that process for testing and evaluating. If you want to jump in here and share some insight, Laura.
Yeah, absolutely. So as Richard and Jack have been saying, if you're creating an amorphous dispersion, you want to ensure that it remains amorphous. So we'll use techniques such as X-ray diffraction or differential scanning calorimetry to look at whether there is any crystalline API remaining. We'll also expose the samples to high temperatures generally to see if it crystallizes over time, because that's always a risk if you've not got a sufficient polymer loading, or you've got too much drug, that it can recrystallize. So looking at the amorphicity on spray drying and on stability is really important. Of course, looking at the standard powder properties is important part of characterization, depending on your target product profile. So things like how hygroscopic the material is, because the more water it takes on, there's a risk, therefore, that you're going to end up reducing the glass transition temperature of an amorphous dispersion and creating a sticky powder. So protecting samples from moisture can be really important if it's hygroscopic. Things like particle powder size, the particle size of the powders, can be less of a critical quality attribute, I guess, for oral, depending how we formulate it. And I guess later on we might talk about beyond the spray dried dispersion, but you want to make sure that your particle size is controlled, it's not changing, and that it matches what you require. Things like water content, because your water content will link into stability, potential micro risk of a powder, as well as residual solvent. If you've got a poorly soluble that you're spray drying, you're likely doing this from maybe it's ethanol, DCM, methanol. You want to make sure that those are below pharmacopoeia limits after spray drying. And I guess the final thing on the bulk powder would be something like a chemical stability. So ensuring that the API is unchanged, there's no related substances that have been generated. It's quite a gentle process, spray drying. You kind of think that you're exposing samples to high temperatures, they're going to completely degrade. But I mean, we've spray dried biologics as well as small molecules. We've done proteins, peptides, oligonucleotides, and it's actually quite gentle. So you don't tend to generate too many impurities from spray drying, but it's always a risk, and its compatibility with the polymer can be important. So I guess they're kind of the bulk powder properties, but we want to make sure that we've created an effective dispersion and that we've created something that will improve solubility and potentially improve bioavailability. So our main standard way of testing these is to look at something called microcentrifuge dissolution. So in this, we'll take a really small amount of material, because we know APIs are really limited at this stage of development. So we'll take a really small amount of our spray dried dispersion, prepare it in really small volumes of buffer, potentially pH, an acidic pH and an intestinal pH, and look at how much drug is in solution. Have we created something that is super saturated? As Jack said earlier, have we got that spring, that improvement in solubility, and are we maintaining that? So we've got an effective amount of polymer. So we'll look at that to screen formulations. We'll look at the intrinsic FESIF and FASIF solubility. I know a lot of people will be targeting solubilities above like 100 micrograms per mil. So that's generally a development target. So we'll check if we've got an improvement in intrinsic solubility. And then probably one of the important ways to understand the rank ordering of different formulations would be to do some sort of in vivo animal model. So that PK evaluation to see how it's comparing to the IV. How is your bioavailability? Is your solubility generating an actual improvement in a response in an animal model? So that is an important part once you've sort of screened down and got your options down to take it forward.
I love it. I think it's going to, Richard, not to say that it's a one-to-one transition here, but I know we wanted to talk about from a DMPK perspective, the approach to designing an oral PK study for spray dried formulations. And I think, Richard Weaver, you're the perfect panelist here to answer that line of questioning. So I'll toss it to you to talk a little bit about the setup for those studies too.
Yeah, sure. And I guess the first thing is, if somebody wants to do a spray dried formulation, exactly as we've heard already from Laura, Jack, and Richard, that there's probably an issue here. Somebody's identified an issue which starts with the poor solubility, and it may also have poor slow dissolution, which has ended up with a scenario, probably preclinically at this stage, that a substandard bioavailability. And then the spray drying has been adopted to try and improve that bioavailability. I'm going to answer sort of that question. I don't want to sound like I'm ducking it, but I do want to talk about the bioavailability bit for a second, because it's a multi-parameter process that's dependent on so many different things. So one factor may or may not change the bioavailability. So the first question I ask as a DMPK scientist, and when hopefully we're going to get on board very early with people like Richard, Laura, and team, is what is the reason for the poor bioavailability? Is it the DMPK reason? So that's my area of expertise. Is it that as it gets absorbed, it's maybe absorbed very well, but it's metabolized very highly by the GI enzymes? So these can be cytochrome P450s and other, or the major drug metabolizing enzyme in the body, which is the liver, which metabolizes 60 to 70% of all drugs. Now, if that's the reason for the poor bioavailability, then actually spray drying won't help in that scenario. And it's important that the customer doesn't get, false hope, let's say, that spray drying is going to be a magical solution. I just wanted to caution that it's not always going to work if it's a DMPK reason, but often it isn't, and there is a way forward. So if it is to do with poor solubility or poor dissolution, then spray drying will absolutely help. Nearly always, I would say, in that scenario. So in terms of designing a study, what one would do is you perhaps have the compound as an HCl salt, or just in the form that it's been synthesized. You would then make the spray dried version and essentially do them head to head in a PK study. And this is preclinically. Obviously, it's far more cost effective, very quick to get this. You can do that in rodents or non-rodents. And you would need to choose your dose level as well. So that's another thing to talk about. So when you choose a relatively low dose level, obviously then the local concentration for an oral study is going to be smaller than if it was a large, a high dose toxicological study, for example, or a high dose efficacy study. So you want to have some confidence that as you increase the dose, this improvement in making a spray dried material is going to translate through at all doses, that it's dose linear. And that's really what you would like to see at some point. So not a single dose level, but a multi-dose level. So I've designed a simple study head to head of your non-spray dried material. But ideally, at least 2 dose levels, and very quickly you will get an answer there as whether this has helped or not.
That's fantastic breakdown, Richard. I really appreciate it. I want to make sure, does anybody else want to add to that? We have a question in there, but want to make sure, does anybody want to add to that?
Yeah, I was just going to say, Richard, presumably to do these sorts of tests, you don't need a lot of spray-dried powder or spray-dried drug, do you? You can go in with relatively small quantities in small animals and get quick data.
Absolutely, and I think this is the important point to sort of make here. I absolutely agree. So you may have seen, or there's lots of papers around there that show animal bioavailability versus human bioavailability, and it looks like looking at a starry sky. There's no correlation there at all, but there shouldn't be. As I mentioned, it's a multi-parameter process, and there's many differences between the animals and humans. But when you sort of take it down into the individual points, the fraction absorbed, something called the fraction escaping gut metabolism, the fraction escaping hepatic metabolism. If you then realize, as you say, Richard, that it is a solubility, is a dissolution thing, then you can do, a rodent study, for example, a rat study, for example, which uses milligrams of material. Low milligrams, 10 milligrams total, perhaps, including running the bioanalytical as well. So as long as you break it down into those individual components, the rodents can inform very highly for those higher species bioavailability checks. So absolutely. And again, to gain further confidence, you could perhaps do it in a higher, more relevant species, which obviously would require a little bit more compound, but still in that milligram region rather than grams. Absolutely. Yes, a very good question.
I love it. Well, I know we're gonna transition here in a moment just to talk about the growth within the area, but I think, Laura, we did get a question in just around the scalability of this. And I think looking at this from early stage development to clinical to commercial manufacturing, how scalable is the spray drying process? Laura, I'll toss that to you, and then anyone else feel free to jump in as well.
Yeah, absolutely. So we're often working with milligram amounts of API in the R&D lab. So we could produce a spray dried batch on our equipment of maybe 500 mgs of solids, half a gram. Ideally, we'd have a bit more. You might get more material back. That's probably the least that you could be working with. So if you're at a 10% drug loading, you kind of need 50 mgs of API in that input batch. But what's really good about spray drying is it really translating process in terms of scale. So here at Upperton, we have benchtop spray dryers where we'd go up to 100 grams. We also then have pilot scale batches where you'd be looking at a couple of kilos. And we've translated tens of 50 to 100 different spray dried projects from that very small scale through. Particularly with oral formulations, where maybe particle size, it's less around the process in terms of particle size control. And we're going to talk about nasal later and maybe how that's slightly different. But yeah, you can scale up to what we have here is a mobile minor Niro, so a couple of kilo batches. And what's great is that, as Richard said earlier, spray drying's been around 150 years. You can get 6-story high spray dryers where you're in with a shovel. So you can really take it up from the bench, the pilot scale, a kilo, to tens to hundreds to tons of spray dried material. And it's the same process throughout. You're using the same technologies as such, the different kinetics, the different steps of the process. So it is really translatable. And then you want to take that bulk spray dried material and make it into whatever product you want at the end. You end up with a nice bulk that you can take to whichever platform you want.
Yeah, I'd agree with that. I mean, the scale up from milligram to gram to kilo scale has been well trodden here and at other companies as well. So generally speaking, that's one of the advantages of spray drying, that you can go up in scale relatively quickly and relatively easily, and the speed of the process as well. You can go from liquid to powder in seconds, and you can make relatively large quantities of spray dried powders in minutes if you want. So it's that scalability, I think, which sets spray drying apart for making these amorphous solid dispersions.
Yeah, absolutely. And I've got another question in here that just came in on top of that one, and I'm just going to ask it. Whoever wants to jump in, absolutely can. This is from Gaurav. He says, "Is spray drying? Can spray drying can be done by fluid bed processor? What is the differences between spray dryer versus fluid bed processor?" Kind of open-ended question, but if anyone wants to answer that, if not, we can continue on. But wanted to throw that question out here as well.
I think there's some key differences between some similarities with a fluid bed dryer, where you're spraying onto a core and you're drying on the surface of other materials like a fluid bed dryer. The slight difference is with spray drying, you have a liquid and it dries to give you a discrete particle, or from a discrete droplet. So the drying kinetics can be slightly different to fluid bed coating, where you're drying a liquid onto a surface. It may not then dry as quickly as it does during a normal spray drying process, but it's a good question, because in many ways the two processes are linked, and there is an element of rapid drying and an element of removal of solvent very quickly to give you a dry powder. But sort of the process and kinetics, I think, are slightly different. I don't know if you'd agree with that, Laura.
Yeah, definite similarities and differences. They kind of leverage on the same base technology. But yeah, slightly different applications.
I love it. Well, it's a great question, and it's a good reminder at the halfway point that if you are still watching, I think we've got 32 people in the session live right now. You can ask your questions directly of the panelists. This recording will be live on the Upperton Pharma Solutions website and on the LinkedIn for the foreseeable future, so you can come back and watch, ask those questions again. But I do want to transition here a little bit, and Richard Johnson, you mentioned earlier, and I can sense some excitement and energy around nasal drug delivery and it being a growing area for spray drying. And I kind of just want to start there. Can you talk to us a little bit more about that growth and maybe some of the reasons for optimism, the advantages I know we'll talk about, but want to start with just that direction in general?
It's a really interesting question and a really interesting part of our business. I'd say up until about 2020, 2021, we'd only ever worked on one nasal drug delivery project. And then since then, the interest in nasal drug delivery has grown phenomenally to the point now where something like 30 to 40% of our inquiries from customers are around nasal delivery. And that's really interesting. And it's been fueled by some very recent product approvals in the nasal drug delivery area. Now, traditionally, with nasal drug delivery, companies or practitioners were developing liquid formulations that you could use a liquid spray and spray up the nose and treat the surface of the nose itself. And generally, a liquid solution can do that very well. It's a very simple liquid spray. But then in the last sort of 3 or 4 years, there's been a transition and a growth in interest in actually delivering drugs into the systemic circulation. So beyond the surface of the nose, into the bloodstream, into the systemic circulation. And that's really revolutionized nasal drug delivery to the point where now people are looking at nasal delivery as an alternative way of quickly getting a drug into the systemic circulation via the nose and avoiding that need for example, for an injection. So that's sort of helped in that growth. But there is one main challenge, and that's because in a liquid formulation, that's fine if your molecule is very stable and it can sit in solution and you can just spray it when you need it. But there's a significant subset of molecules that are not stable in aqueous solution. And I'm particularly thinking about some of the larger biologics we've mentioned earlier, molecules that are unstable in the aqueous environment. And one of the best ways to stabilize those larger, more sensitive biological molecules is to make them into a dry powder. So by drying them, removing the water, you can stabilize them. But to deliver into the nose, you need not just any old dry powder. It needs to be a certain size, a particle size that you need to get the drug or get the biologic into the nose needs to be within a set size distribution or size range. And that forms perfectly into the spray drying remit. That ability not only to make a dry powder, but to stabilize the drug within it, and engineer it into a particle of the right aerodynamic size and properties to be able to spray into the nose, deposit in the nose. And importantly, there are no small particles that can then potentially go through the nose into the lungs. So coming back to that initial concept I mentioned earlier about particle engineering, so clearly spray drying is by far the best technique for producing dry powder formulations for nasal delivery, either at the small scale or much larger scale. And really that's fueled that growth in interest in spray drying and nasal drug. I've got to get spray drying in there. It's a great technique and it fits perfectly within this area of application.
Well, and just to say out loud, I love I mean, it's very clearly how excited you just became when I asked that question. And it also then led to a flood of different questions into the LinkedIn Live. And so I'm gonna press pause for a second and ask, we've got 2 really good questions from 2 different individuals here. The first is from Jock over at M2M Pharmaceuticals, that it's a question about if you spray dry without polymer, so water insoluble API on its own, do you expect to get amorphous or crystalline spray dried material? And so I know this isn't exactly where we were headed, but I want to ask these couple of questions as they came in as we've got this group here. So want to toss that one out. Whoever wants to answer it, we can create a conversation around that.
Well, I was going to say that we've seen a lot of projects where maybe you're not just using spray drying for particle engineering, where you get a combination of the many utilization of spray drying. So where you're creating an amorphous dispersion by spray drying, making sure the particle engineering means that your particle size is suitable for nasal. And you can follow the same techniques as for oral, as long as you're using precedented excipients that are suitable for the nose. then you can create an amorphous dispersion that's got rapid dissolution at the nasal mucosa when it's administered. So we see a lot of projects where you're looking at poorly soluble APIs, spray drying, so that you get fast dissolution at the nose. So whether a product is amorphous or crystalline on spray drying will depend on the excipients that you often spray dry it with. So certain excipients on spray drying will remain crystalline, and that might keep your drug crystalline, or they may turn amorphous. So your excipients can influence that, but we definitely get a lot of overlap. So we have many projects that are poorly soluble that we can create really fast dissolving at the mucosa, maybe include mucoadhesives to help it retain on the surface. So yeah, that combination is really something that we've seen a lot of, Richard, haven't we?
Yeah, I'd say so. But you know, just to preface that, I think from the original question, there was that question of if the drug doesn't dissolve in an aqueous vehicle, for example, and you spray dry, you can still produce a dry powder. But if the drug hasn't dissolved, then presumably it'd be in the same physical form that it went into the aqueous suspension. So if it was crystalline in aqueous suspension, you then remove the water, then I suspect that drug is going to primarily stay in its crystalline form. That's not to say you can't improve the bioavailability by just dispersing it and retaining it on the surface of the nose, for example, could certainly improve the bioavailability from a dry powder compared to a liquid spray.
I guess to add to that as well, if you were to spray dry it from a solvent that could dissolve the API, then there is a small possibility that you could form an amorphous API from the previously crystalline API, but that would be very much dependent on the properties of said API. And generally, without any sort of stabilizing excipients there, we would typically expect it to revert back to, I guess, the most stable form, which would be the crystalline form.
I love it. I love it. Okay, it's a great question. Appreciate that one coming in, and a good point here that you have access to the experts, which is what we'll do. Again, we've got a question here from Emma over at DFE Pharma, and the question is, from your experience, what are the most useful tips, tricks, things to know to overcome or minimize the stickiness problem with spray drying of sugar solutions? And just going to open this one up to the crowd again, if anyone wants to jump in and answer Emma's question there.
Okay, I'll perhaps have a first stab at it. It's certainly a good question for certain types of sugars. If they have a low glass transition point, then stickiness can become a problem, both from from the glass transition point of view, but also from a general hygroscopicity point of view, where you're going to reabsorb water back into the sugar as it's forming a dry powder. And clearly, one of the ways around that generally are to move towards, low temperature spray drying is one thing that we do. We don't need to work at high temperatures typically. We may need to drop down the drying temperature, the outlet temperatures, to bring that temperature down below the glass transition point of the sugar in question. The other thing you can do is add other excipients into the sugar formulation to bring the glass transition point of the powder back up. So it is a challenge, and certain sugars do form sticky complexes or sticky powders. But by controlling the temperature, reducing the outlet temperatures and drying temperatures, and also the addition of additional excipients to raise the glass transition are two things that we can do routinely.
Jack, I see you nodding your head. Is that an agreement mic drop, or do you want to add anything else here?
No, I guess I completely agree with Richard. Yeah, it is, I guess, 2 points at which these formulations can become sticky, as it were. Firstly, during the spray drying, and Richard's largely addressed that, but I guess it can also become sticky on storage and handling as well. So, I guess taking good care, handling under low humidity environments, ensuring the formulation is well packaged and away from moisture will also help protect the formulations and stop them sticking.
Wonderful. Well, we've got a a sound of appreciation from the two questions there, and thanks for answering both of those. We've got about 20 or so minutes left here in our conversation. Please continue to throw your questions in, and if we don't address them today, I promise that the Upperton team will be able to answer them. We'll put some commentary out as a follow-up. So please continue to put those in. Richard Johnson, I'm gonna come back to you. We were kind of going on this pathway on the energy and the growing area within nasal drug delivery, and we just kind of started to talk about the unique advantages and the challenges. I want to make sure before we go into performance and characterization tests that you feel like you were able to address the unique advantages or challenges of spray drying for nasal dosage forms versus oral. I know we kind of touched on it, but I want to make sure if you have anything else to add in that area, that here's the opportunity to do so.
Yeah, I can certainly add a bit more into that area. I mean, here at Upperton, as I mentioned, nasal drug delivery has become a big part of our business, and as part of that sort of growth in the nasal drug delivery area, we've developed a platform called the Upper Nose Technology. It's a screening platform where somebody's can come to us with their molecule, and we'll then look at that molecule and develop the best nasal dosage form that's required to deliver that drug into the nose. It could be a single dose, it could be a multi-dose, it can be a liquid, or it can be a dry powder formulation. But today, obviously, we're focusing very much on that dry powder, and there's some unique challenges and advantages of spray drying and some challenges that come with it. The most important thing we do in those early development stages is to find the right excipients to help formulate those drugs that we're going to deliver. It could be a biologic, it could be a peptide, a protein, it could be a small molecule. But we want to produce a dry powder in which, first of all, the drug is stable. So we need to have good compatibility between the drug and the excipients present in the solution that we're going to spray dry and in the subsequent spray dried powder. But also, once we've made those particles, those spray dried powders, those early formulations, we need to look at the stability, because there's no point in producing a spray dried powder for nasal delivery if after three days or 3 weeks the drug falls apart, or it interacts with the excipients, or there's aggregation or breakdown of the product. So in those early stages of the Upper Nose platform, we do a lot of screening where we make sort of pilot formulations with drug, with different excipients, and we spray dry them and produce particles and look at the stability and the performance to make sure there's no damage and that the drug and particles remain stable. That's one big part, is that stability challenge. The next big challenge is all around making particles of the right size, because as I touched on earlier, we're going to deliver these powders into the nasal anatomy, into the nose, using a device. So really early on, we want to be able to spray dry and make particles of the right size for nasal delivery. And again, can be quite a challenge, particularly in an early stage in development where you're working with relatively small quantities of drug to make big particles. I'm talking of particles in a sort of 10 to 50 micron size that can go into the nose but will not then subsequently pass through into the lung. So coming back to the approach and some of the advantages and challenges, clearly spray drying is all around the advantages of particle engineering, getting the right excipients and then making particles of the right size for delivery into the nasal anatomy. And that's a process we've done many times with lots of different types of drugs. And there are certainly a number of challenges and hurdles to overcome to be able to do that.
I'm glad we went back to that, because it seems like something that we can discuss, and it's also kind of a good segue into that. What kinds of performance or characterization tests are important at this stage? And I know, Richard Weaver, we want to talk again about when it comes to nasal PK studies, how does it differ from the oral side of things? So this is for both Richards, and I think it might just turn into a conversation between you two, but looking to just transition from the kinds of performance or characterization tests are important, looking at PK of nasal formulations, and then how does that relate to the setup of or approach to the PK studies? I think Richard Johnson will start with you, and then maybe pass this on to Richard Weaver as we work through this too.
Yeah, so probably one of the final pieces in the early stage jigsaw, if you like, is, as I've mentioned, making particles of the right size. Now we can size particles using something like a laser diffraction equipment, like a Sympatec, for example, which will give us a size distribution. And we know if we're in that sort of 10 to 50 micron particle range, that's where we want to be. We can also test the chemical stability to make sure the particles look robust and stable, the drug remains stable. And then the next stage before we pass on to Richard and to his team is to look at how that powder then works in the device. So to get the powder from the container into the nose, we need a nasal device. So we can load that powder into a nasal device and fire the powder into an artificial nasal cast. We use something called the Alberta idealized nasal inlet. It's actually it's designed to mimic the geography or the anatomy of the human nose, and we can fire that powder that we make from the nasal device into the idealized nasal cast. And the powder will then deposit in different parts of the nose because the nose is made up of different compartments. So that's a really important last test to show that we've made particles of the right size, the product is stable, it remains stable, there's no chemical damage to the drug. We can then look at the performance in vitro in this nasal cast, show it comes out the device and deposits in the artificial nose. So that's great groundwork, if you like, to then be able to go over to Richard Weaver and his team and say, here we have a powder, looks like it's got the right physical and chemical properties, but hey, how does it perform in a real model, such as an animal model?
Yeah, that's a great introduction to that then, Richard. Yeah, and the first question that what I would ask as a DMPK scientist or try and understand is why is the customer trying to deliver via nasal route versus say an oral, an IV or subcut or whatever in this small molecule space. And often people choose intranasal for fast onset. Okay, so the high capillary, this high surface area generally. When something goes into the nose, it does actually look quite like an IV injection. It's the blood concentration gets in very quickly and comes down in a typical IV fashion. So fast onset is one thing. Trying to maximize the bioavailability as well. So we already talked about the multiple hurdles of trying to get good bioavailability from an oral for an oral drug, and there are so many barriers there. It's got to survive the low pH of the stomach. It's got to get into the GI tract. Plenty of enzymes in there. It's got to be absorbed across across the intestine. Survive the drug metabolizing enzymes there, etc., etc. So often, excuse me, I need a drink of water. Sorry. Hopefully, you can filter that one out. Yeah. So it's often nasal. Or bioavailability is generally, is very, very high usually. Obviously, it can have a big range, but there's a big increase generally in bioavailability. And the way you would do it preclinically is to actually do it head to head. So you would take your standard IV dose, which the bioavailability is always 100% from an IV dose by definition. You'd potentially have already existing oral bioavailability from that compound, often, especially if it's a repurposed oral drug. And then you would look at the bioavailability from the intranasal routes as well. And does everything work? Does everything hang together? Is it improved bioavailability, intranasal, or versus a different formulation from an intranasal perspective? Do you get that fast onset if that's what you want? And what is the bioavailability? You can calculate that out. So the other thing to say as well is often, it's obviously intranasal is device limited in terms of drug loading. So often you don't give as much. So generally these compounds are higher potency than an oral drug as well.
Fantastic. Richard, I just want to make sure we're playing ping pong between the two Richards here. I want to make sure you don't have anything else to add on your side before we move into our final line of questioning here.
No, I think that's a perfect introduction and explanation from Richard there. And I think what's been most interesting and exciting from me over the last couple of years is the number of drugs that are now coming onto the market and seeing this utilizing this fast delivery, this fast onset of action from the surface of the nose into the systemic circulation. And most recently, Neffy, for example, epinephrine, which is normally delivered by intramuscular injection, somebody suffering from an anaphylactic shock, they use an EpiPen. You can now deliver the same dose of epinephrine via the nose. And in actual fact, it gets into the systemic circulation quicker by the nose than it does by an intramuscular injection or subcutaneous injection, which is quite extraordinary. And I think that's piqued a lot of interest now in the pharmaceutical industry that, hey, this delivery into the nose is a real potential alternative if you want a fast-acting drug, if you want to get into the body quickly and avoid that need for an injection. Self-administration of a nasal dose has a lot of pluses and advantages.
Also, in terms of patient as well, patient experience, intranasal delivery versus, a self-administered intramuscular, it's pretty clear which is the best way forward here. It's worth mentioning that.
Yeah, no, absolutely.
Yeah. Okay, 50 minutes in, 50 minutes of fantastic conversation. Laura, I'm going to go to you for the last question here before we get into some wrap up, next steps and tidy this up with final thoughts. I think, Laura, the question here is kind of to tie all of this together, right? To bring it together. Once a lead prototype has been selected, can you talk briefly about what are the next key steps to take the product forward to kind of tie a bow on a lot of this conversation?
Yeah, of course. So for nasal, sometimes the spray dried powder itself is what's filled into the device. So as Richard Weaver said, often potent molecules, you've got your dose in 20, 30, 40 mg of powder. So really it's quite straightforward. It's filling it into devices, generating your stability and hopefully onto some clinical data. So you could just fill that powder as a bulk into the devices. And device selection is really important. We do see that the target product profiles in terms of what your particle size on spray drying needs to be is different according to whether you've got an active or a passive device. So it's quite straightforward for nasal, really. And then it's the characterization there. When you're looking at, say, oral formulations, there are different ways that you could administer that ASD. So the most simple way is probably to do like a spray dried dispersion in a bottle to clinic. So just creating a suspension, giving it to the clinical trials and generating your SAD/MAD data. People might like to fill their spray dried dispersion straight into a capsule for administration. The issue there, sometimes ASDs can kind of clump on wetting. They don't disperse very well. You do generally need to formulate them to generate the best dissolving powders. So people sometimes want to just fill ASD in capsule. You've got to make sure that it comes out of the capsule if it was taken orally. So I guess you can then just take your ASD though and put it through conventional manufacturing techniques. So you could granulate that material to create a higher density, better flowing granule that you can then tablet or fill into capsules. And then you're kind of onto the conventional approaches for formulation development, really, generating your stability, manufacturing batches for tech and through to clinic. So you can then take your ASD, put it in a tablet, and there are several ASDs already on the market, a lot of antiretrovirals that have been spray dried for the last 20 years. So there's a real track record of spray dried dispersions in commercial products, and we're really seeing plenty through the pipeline that offer that bioavailability enhancement that can go through clinic just as standard tablets and capsules.
Fantastic. Anybody else want to jump in on that? Want to give reason for or give an opportunity for that? Okay. Well, I have a surprise segment here for the team. You guys did not know this was coming, but it is one of my favorite features at the end of our LinkedIn Lives here. I'm going to put each of you on the spot. I've got my handy dandy timer out, and we're going to ask for 60 seconds, just final thoughts, takeaway. Hit us with something that you are optimistic about looking forward, something that you view as the reason that someone should consider this. Wherever you want to go, I want you to give us that 60 seconds. You can think of this as your sound bite that we will utilize following this episode. And Jack, I'm going to put you
Let's go to Jack first.
I'm going to put you on the spot first. I won't stop you at a minute, but we will shame you if you go way over the minute. That's the rules. And so Jack, whenever you are ready, your time will begin for your final takeaways here from our conversation today.
I guess it's mainly around just how powerful a tool spray drying can be as a bioavailability enhancer. As we've discussed, there's so many development APIs out there that are coming through that are, poorly bioavailable or, just lack solubility. Spray drying offers a real, unique opportunity to improve these solubilities. So, we have a lot of options available to us. We've discussed, using polymers to produce ASDs, but there are also other options we can undertake as well. We can look at formulated with cyclodextrins to produce inclusion complexes. We can also include other excipients within these formulations as well that, also enhance bioavailability. So, not only do we have a better solubility API, but things like TPGS as well can also improve permeability or inhibit efflux of the glycoproteins. So, there's the world's your oyster, really. You can, mix and match your excipients to develop a profile as you wish. And I think that's the beauty of spray drying, is the versatility.
Really impressive. Being on the spot 1 minute and 8 seconds. That's not bad. That is, everybody else, there's a standard that has been set, which is really important because Richard Weaver, you are on the clock, and whenever you are ready, we are just looking for that final takeaway from today's conversation.
Yeah, no pressure then, eh?
Exactly.
No, I think as an overall thing, getting a drug to the market is really difficult. We're in a very high attrition industry. And from my view, the most successful teams are the ones that are multilingual in the other disciplines. And there's so many disciplines, in research and development. And I guess something I'm just super passionate about, and this is, thankfully why I've been invited today, is that link up between DMPK, the CDMO, and that sort of formulation development, pharmaceutical development, if you will, of how important it is to talk together as the same team with the project team, whoever they might be, the biotech. And that's really my take. I've seen programs as one, I can't talk about the molecule because it was a customer compound, but it was taking a compound that had 2% bioavailability that went to over 90% bioavailability with a spray dried dispersion. An amazing result, and it was dose linear as well. And that was only possible because we understood the DMPK properties of the molecule and realized it wasn't a DMPK reason for the poor bioavailability. It was down to poor solubility and poor dissolution. So just to say, when these heads are together, there's a lot of success to be had. And I'll stop there, and hopefully it's not over 1 minute.
I love it. 75 seconds is not bad at all. And I also love, the thought process around this has been such a technical and specific conversation, but bringing it back to that core reason of getting a drug to market is really difficult. And the collaboration that has to be in play is super essential. And so really appreciate that Richard. A fantastic takeaway, just as yours was, Jack. And Laura, that means, number 3, you're on the spot here, and then Richard, you'll be last. So whenever you are ready, Laura, all you.
Yeah, I think I wanted to echo what Richard Weaver said. So I think it's really understanding your molecule. Though we would never say, we've talked about spray drying for 50 minutes, but we'd never spray dry if you don't need to. Like if it's a different reason for your bioavailability issue, you need to understand that, because there's no point spray drying it, creating something more soluble, and it's not going to be any better in the clinic. And likewise, there might be molecules that you can look at micronization first, or can you develop a formulation that's got a pH modifier in it, and that's going to improve your solubility. So don't just, maybe people, I always like it when people just say, right, understand, figure out what you need to do, come to us with the different options. And we're so mindful of the commercial considerations, the patient considerations, the technical, what you're working with. So that's what we really enjoy. We really enjoy spray drying. We do a lot of it, but probably 50-60% of our projects are not spray dried because there's no need to. So I think it's looking at it with fresh eyes. That's what we enjoy, and that's why we enjoy working at Upperton, I guess. And in a CDMO space, we can bring the experience from other projects and leverage like the most chance of success of taking that molecule to market.
I love it. 72 seconds.
Ugh.
We're fantastic. And I also love that concept. You know, again, like you said, we talked a lot about spray drying. We talked a lot about this specific area, but the challenge, the complexity, and at the end of the day, the patient considerations, that is at the forefront of everything that's being done. So I really love that, Laura. Richard, you know, the microphone is yours to tie a bow on this conversation. Whenever you're ready, it's all you.
Yeah, okay. I think I'll focus primarily on the nasal side of things. And really, I've been really super excited to see where nasal drug delivery, and in particular, spray-dried formulations for nasal drug delivery have really expanded and grown over the last sort of 2 or 3 years. And as I mentioned, it's primarily around target. First of all, a realization that the nasal pathway is such a great way of getting drugs into the body quickly, I think has really opened up nasal drug delivery. And spray-dried nasal formulations are an important part of that. But also, I think one thing to bear in mind is that when you go into the nose, there are different parts. It's not just about spraying a drug or a biologic, for example, as a dry powder into the nose. You actually get different effects in different parts of the nose. So, for example, if you're developing a vaccine for nasal delivery, it's a huge growing area now. There are actually different parts of the nasal anatomy you want to target, such as the nasopharynx, where most of the local immune response will be raised. If you're looking to target your drug into the systemic circulation, you're really looking to target probably around the turbinate regions, the highly vascularized regions in the nose. And finally, one of the most interesting and fast-growing areas is what they call the nose-to-brain targeting, where you can spray a drug into the nasal cavity and it can hit a certain part of the nasal anatomy called the olfactory region, which is the only part in the human body where the brain has outside access without going through the blood-brain barrier. So now people are looking at that nose-to-brain route as a way of getting a whole new raft of treatments to treat various brain diseases such as Alzheimer's, such as various cancers, Parkinson's disease. Drugs that normally can't get into the brain can go through the nose. So the take-home message from me is there's so much more we can learn and understand about nasal drug delivery, and having a dry powder formulation is really going to be at the forefront of that in the future. That ability to stabilize molecules, sometimes much bigger molecules than you would normally expect to go into the nose, and we can get them into the body through these different routes in the nose. So it's clearly a fast-growing but a very interesting area that I know is going to really sort of shape the future of drug delivery in many areas.
I'm not going to tell you how long that
Hey.
was. Not even going to say it because it was a
bang on? It was bang
fantastic.
on, wasn't it?
It was, listen, 2 minutes and 28 seconds, but that was a perfect end, and it segued into my final takeaway. So you can steal my minute, which is the future of where we're headed. And there's clearly a lot of reasons for optimism that we're going to be able to solve challenges utilizing this formula that we weren't able to do before, and that Upperton Pharma Solutions is uniquely positioned to be that partner for this going forward. And so I just want to say, Richard, Laura, and Jack on the Upperton side, thank you so much for participating in this conversation. And then Richard Weaver over at Drug Discovery Solutions. This was a fantastic discussion. Clearly a lot of scientific expertise, but also real world examples and actionable insights for those listening. And if you are listening to this as a recording and you have questions for anyone on the team, even if it's Richard Weaver over on his side, fire those questions into the comments and we will make sure that we are able to answer those questions. And then if you watch this and you think that there's more content to be had, just reach out and we'll make sure, maybe we'll do another one of these in a couple of months, because I think that you guys did such a fantastic job. So really appreciate your expertise, and to everybody that watched today, thank you so much for sharing your time and asking the fantastic questions that you did as well. So on behalf of Upperton Pharma Solutions, thank you so much to the panelists and to anyone watching, and I hope everyone has a great day.